But I want to do it. Right? So it doesn't matter which one, but I like that. So. 21st, Wednesday. Oh, yeah.
Hi. Hi. Ah, é? Você é professora aqui?
Não, eu sou de aposentar na USP, Federal. Meu noivo é daqui. E agora a gente vai fazer.
Mas você, como é que você soube?
Porque eu venho e eu vim semana passada e ele trabalha. Não, ele trabalha, ele é cientista também, estou fazendo assim, mas eu estou fazendo tudo mais. Tem que fazer essas coisas aí.
Claro, obrigada. A minha chefe é brasileira também. A Daniela é brasileira, ela vai estar aqui, eu acho. Acho que ela está lá no laboratório. Vai, bom. É.
Então, mais uma.
Obrigada.
All right, Anna, Caroline, Caroline.
In my language, Ana Caroline. But you can say Anna Caroline, that's fine.
No, let me try.
Ana Caroline.
Anna Caroline.
Yeah.
Gandara? Gandara?
Gandara.
Gandara. Anna Caroline.
Gandara. But I don't mind if you say Anna Caroline.
He might speak some Portuguese.
Yeah,
Yes.
she just came.
Nicely done last week. Thank you. Yeah. This one, probably changes since. Yes, to myself. I will. It's just.
Yes.
That. But these are this, so these are college. Yes, they are. Why is it? Because you're here every day. You know what I can do? A lot of people like it. COVID swab. Yeah, do you remember the no, the pen, John.
That's why women have those long hair lines. It's that's because of the hair and they needed to, they Yeah.
There we go.
Thanks.
Yeah. So it is quite fun to get hot water as a cold water. Yeah.
Oh, I'm good. Thank you so much.
Yeah, I like the CBS. Docs like it.
You know, I used to bring my kids here and I started over they're 20 now, no. It's 20 some years old now, they said, do they still the pencils? I said, no, but now they do.
Well, because that's my weekly message.
Oh, okay.
So Eric, tonight? Toronto. Oh. So we're gonna have to drive.
Were you yeah, fine, so I am.
So that is just on the back is about.
Oh, okay.
Advanced United Workers.
Oh, okay.
What is and chapters? Oh, how about that? So we don't really. Probably this way. Go on. It's the back door. Is your microphone on?
Yes, it is.
Okay, we're gonna do a sound check. Can you count to 20,000 slowly? 1, 2, skip a few, 20,000. How does it sound for you?
I mean, what they say.
You
I
have to
think it's
speak.
fine. Is that fine? Want me to speak louder? I can speak louder.
All right. Well, thank you, everybody, for coming out tonight. I'm gonna do a little informal version and then we'll kick into the formal version. I'm gonna ask you 5 questions. You will know the answers.
Okay.
Thanks for coming out tonight. Next week, September 21st, Michael Menon from the Bureau of Aeronautics in the Wisconsin Department of Transportation will be here to talk about at the intersection of wildlife and aviation. So he studies the wildlife ecology of animals that hang out on and near and above airports. Yeah. On September 28th, Lindsay Snyder from the School of Veterinary Medicine will be here to talk about anesthesia and pain management and veterinary care. And she does some work with acupuncture, which I'm going to be very interested to see how that works with animals. On October 5th, Johanna Oosterwijk from horticulture will be here to talk about vertical gardening. So if you've been reading about how we can grow plants at a commercial level vertically in plastic containers or otherwise, It's a very cool version of hydroponics, but she's going to give us the math on all that. So thank you for that. Now you get to come over here. Let's see both of us. Oh, perfect. You waved. You're not supposed to wave.
No,
You broke the fourth wall. Welcome, everybody, to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for the Division of Extension Wisconsin 4-H. And on behalf of those folks and our other co-organizers, PBS Wisconsin, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. Tonight, it's my pleasure to introduce to you Ana Caroline Gandara. She's from Brazil.
yes.
She just made me fluent in Portuguese. Maybe not. She's going to talk with us about climate change and the consequences of suboptimal temperatures on reproduction. I get to ask you the 5 questions.
Okay,
Ana, where were you born?
Brazil.
Where in Brazil? It's a big country.
In Rio de Janeiro, Brazil.
One of the 4 places I've heard of. Great.
What are the other ones?
Paraná,
Paraná, okay.
Sao Paulo,
São Paulo,
and Rio de Janeiro.
Rio de Janeiro, okay. And the 4th one?
Never mind.
Yeah. Don't say Buenos Aires.
Yeah.
Where does Montevideo come in?
Yeah.
Okay, Santiago. Okay, where did you go to high school?
High school, a private school called Colégio Santa Mônica.
And where'd you go for your undergrad and what did you study?
Oh, actually, no, you made me confused. No, my high school.
My fault, my fault.
Why did I thought, why you are asking this? Actually, I went to a polytechnical high school, so I have a biotechnology degree. I could work in a lab as a technician
Very good.
because of this degree.
And then where'd you go for your PhD and what did you study?
I have a PhD in biochemistry at the Federal University of Rio de Janeiro.
Good. And when did you come to Madison?
Because the whole lab moved
Mhm.
from Baltimore, Maryland. So we were at Johns Hopkins University and my PI moved and everybody came.
What year was that?
I'm not sad, actually.
No, what year was that?
When?
Yes, thank you.
July.
Of
Last
this year.
July.
Last July.
Yeah, we are here.
It's very amazing. That's quite the saga.
It was.
I'm looking forward to your talk.
Thank you.
Suboptimal temperatures on reproduction. That's important.
Yeah, I hope it's going to be more fun than watching Netflix, at least now.
Setting the bar high. Would you please join me in welcoming Ana?
Thank you. Hi to everybody in the Zone and or YouTube, whatever you're watching, and thank you for. I wasn't expecting this big audience because I don't know, because after pandemics, I don't know how people are behaving very well, especially here. So as I said, we just moved. So yeah, so we moved. I'm assistant scientist at Drummond Barbosa lab. She's here. She's shy, but I will show her in the audience. Actually, we can skip and go to the acknowledgements. Wait. Okay, so yeah. So she's here. So she's the PI, and yeah, we are a small lab. When we were there, we moved. Rodrigo is the scientist. Emily is a grad student. That's also here. Arthur is my son. He's not here tonight. But things actually happened very fast. And now we already have 3 undergrads, 1 rotation student, and a postdoc starting next month. So definitely this picture cannot be used anymore in the next seminar. Thank you, Tom, a lot for this opportunity. And of course, I need to thank the funding. As I said, we moved from Johns Hopkins. We are here at the University of Wisconsin-Madison at the Department of Genetics. And we are also affiliated to Morgridge Institute for Research at, if you cross the street, you're going to see the building. Yeah, so I'm going to start showing this amazing slide that CDC created, showing the impact of climate change on human health. Here we have like the major events that climate change is causing, as more extreme weathers, rising sea levels, increasing CO2 levels, rising temperatures. I forgot what I read it here, but and all the consequences of it. For example, extreme heat is happening, and because of this, we have more heat-related illness and death, cardiovascular failure. But actually, we have all these things happening right now. And that, but I'm going to focus on reproduction. I don't know if everybody heard about the IPCC report. That is the Intergovernmental Panel on Climate Change. That is a report produced by active scientists. It's a voluntary work. They analyze the literature. So it's very important to emphasize that what the report illustrates is data based on facts. So, and last year they released the last version, the most updated. It has almost 4,000 pages. I downloaded. Everybody can download it. Actually, it's public. Everybody can consult it. There is a summary for journalists, for lay public. They have nice graphs and figures trying to summarize this. Of course, I don't expect you to read this, but this is one of the figures showing the effects of heat and cold, more extreme heat and cold temperatures on the planet. And in this case is the regions, the number of land and coastal regions that are going to be impacted by these more extreme hot or cold temperatures. So this is one example of this report. But if you go to the text, specific about these temperatures, that is what I'm going to focus here tonight, you can see that actually these effects are being actually, these effects dictate many aspects of crop cultivation, agroforestry, livestock production, and the involvement of insects in all this. So, as I said, this is all based on literature. So this is already happening. This is not a hypothesis. Talking about heat, more extreme hot temperatures. This paper published in 2020 analyzed data in USA specifically from 1930 to 2010, showing that there is an increasing trend in heat waves. If you go and check this paper published last year, they discussed that the burden of heat-related mortality that is increasing. We have more people dying of hot weather around the world. 37% of these deaths are related to anthropogenic climate change. So this means that human activities are worsening climate change, accelerating climate change on the planet. So, and we can attribute this by human activities. And there are a lot of methods to measure how a human body can tolerate this heat. Many methods, and this paper was published this year, got some attention from the media because they picked this method called wet bulb, that you subject humans, individuals, to warm and humid conditions or hot and dry conditions, and you measure by clinical analysis that I can't talk very well about it, and they concluded that actually the previous hypothesis proposing like 35 degrees Celsius in humid conditions was the maximum temperature a human could tolerate, like healthy. And they said, no, actually it's around 30, 31. And what they are discussing, that's maybe we are underestimating the effects of the hot weather, and maybe we'll suffer like earlier than we are expecting. So 2 slides ago, I just mentioned that there were insects involved in agroforestry, crop cultivation, etc. And that's what I'm going to try to convince you why we need to study effect of temperature on insects. And actually, this is super outdated. These numbers are from like almost 20 years ago, but the insect is the group of animals, the most diverse group of animals on planet. They essentially occupy all ecological niches on the planet. And probably you know that they have essential roles, right? They help in the soil turning, aeration, they are pollinators, they are recyclers, they work as food for the wildlife. Besides, they are disease vectors too. So they are all insects. And still you should say, I don't care anyway. So I'll try to convince you with this slide why we should study. The most romantic argument is we should preserve biodiversity. And you still say, I don't care. Okay, why we should study the effect of temperature insects? We can understand and try to help in the control of disease vectors and agricultural pests. For example, we can help or turn more efficiently the food production using insects as protein source. You think it's gross, but you already eat insects, but you don't know. Shrimps are like insects in the sea. So if you eat shrimp, you eat insects. So it's not that gross. We can understand better insect-mediated pollination. And even if you're not worried about Amazon, that's fine because you need insects to pollinate the crops. So you should worry about it. And last, we can understand better how these organisms are dealing with all these changes that is already happening on the planet. Many reasons. And I will talk about Drosophila Melanogaster, that is an insect, and is also known as fruit fly. And it's a powerful model system that was first used by Thomas Morgan. He established these insects as a major genetic model. This was his seminal work that was published in Science in 1910. And we use Drosophila because it has short generation cycle. It's very cheap to keep the stocks at the lab. It's very easy to manipulate the genetics and the behavior. It has higher evolutionary conservation with humans. So around 75% of human disease genes, Drosophila also has. And we have many tissues and body parts of humans have counterparts in flies. So we can use these fruit flies organs to study something related to the same organ in humans. And also we know, and we're going to see the next slide, that both they have highly conserved biological process that they share. And 6 Nobel Prizes were awarded to scientists that use fruit fly as a model system. The first one was the Thomas Morgan in 1933, because he uncovered the role played by chromosomes in heredity. But we also have contributions of flies to understand mutation rates by X-ray. We have knowledge coming from embryonic development using Drosophila larvae, knowledge coming from olfactory system, about olfactory system, innate immunity, and circadian rhythms, that it was the most recent one in 2017, all using Drosophila as a model system. But we have many other conditions, human conditions that are studied by Drosophila, using Drosophila as alcohol addiction. Yes, the flies, they can get drunk. That's exactly why they use it. Autism, stem cell biology, that's what we do in the lab. Sleep, epilepsy, and go on. We have many, many disease models to use Drosophila. So this is a summary of the first part, just to remember, like, climate change is affecting populations of plants and animals, not only insects, all organisms. Insects are the most diverse group of animals and have essential roles in the planet, and that fruit flies are a powerful model for biomedical research. what I'm going to do in the second part is to show you some information that tries to answer these two questions here. How climate change and its disruptions in temperature affects the reproduction of insects, of temperature susceptible animals like insects, and how human fertility is affected when temperatures are too high. So that's what I'm going to do in this next part. Okay, so any organism is capable to adjust its physiology in response to different stressors. It's not only temperature. So in humans, fertility can be affected by mental stress, poor diet, obesity, excessive exercise. In our question surrounds the effects of temperature, how temperature affects fertility in humans, because humans, despite mammals, not only humans, but mammals can control their internal temperature, right? But even controlling their internal temperature, we still can be susceptible to situations like fever, heat stroke, or hypothermia, where the body is not regulating very well the temperature. So, and these can lead to death or other impairments. And a very common thing that happens in the organisms is remove energy from reproduction to deal with some stressor. This is very common in infections. So if you're dealing with insects, and you see these very well in fruit flies, the fertility and the gamete production will be impaired because they need energy to deal with the pathogen. So this is one example. And we already have data actually about the effect of temperature on insects. And I'm going to show you very, very briefly some data, not mine, also mine, but past data. We know that if we subject flies and larvae to extreme temperatures, like very cold, less than 12 degrees is very cold for the flies. They actually enter in a process called diapause. They slow down they super slow down their metabolism. And the main effect of diapause is block the yolk uptake on the ovaries. And on the other hand, if you subject them to very hot temperatures, 31 degrees, okay, I need to mention that these temperatures depends on the species of the Drosophila, and now I'm talking only about Drosophila Melanogaster. Okay, because we have different species of Drosophila around the world, and they are adapted already to different temperatures on the planet. So I'm talking about Melanogaster specifically, that is this guy here. So 31 degrees is already too hot for them. And we already have data showing some impact on body size, survival for sure, and egg numbers and size of the eggs, and so on. But the key thing is most of these papers were done in larvae, and I'm going to show in this next slide what is that. And we have very few data that were done in adults, that is this guy here. And the very few papers that tested these on adults, they tested these very hot temperatures, actually not this one, 38, 40 degrees. So for a short period of time, and we call it acute thermal stress. And this obviously will impact the fly in actually many aspects, not only reproduction. So for those who never thought about an insect and how they develop, not all insects, but some group of insects, they develop in more or less like Drosophila. So we'll have an egg, and then a larva will hatch from this egg. In this case of Drosophila Melanogaster, it will take 2 to 5 days. It depends on the temperature. This larva will incubate, and this is the stage where the Drosophila is the metabolism is very, very slowed too. So it's almost like a mummy, but it's not a mummy, of course, but it's there working very slowly. And then from this, an adult emerges, and this is the stage that they are sexually mature, and then they can mate and lay eggs, and we restarts again. So I said most papers did all the studies on this stage, but these guys, despite they are the same individual, they have very different physiologies. Very different physiologies because their goals are completely different from each other. This guy wants to eat, to grow and develop healthy, and this guy wants to survive, to reproduce. So different goals, different physiologies. Makes sense, right? And this is the very first paper, as far as I know, that published like the effects of temperature on Drosophila fertility. This was published by Northrop in 1920. And he did a very simple experiment. He raised flies at 22 degrees and got these flies and subject them to different periods of time at 30 degrees. And he checked if they laid eggs and if these eggs hatched. And he saw that doesn't matter how many days they stayed at 30 degrees, they laid eggs, but no larva hatched. So if we could do an analogy to humans, it's like you can get pregnant, but the pregnancy doesn't finish and you don't have a baby in the end. So it's the same. So if you think about from the ecological point of view, you can get pregnant, all the flies can lay eggs, but if you don't have an offspring, health offspring coming from this, the reproduction of the species is impaired anyway. Another paper published in 1932 was done by Alpatov, and he raised the flies in 18 degrees or 30 degrees, and also underfed with no yeast or very few yeast, very few protein in the food. And he saw that the number of eggs laid were different. But the most important thing about this paper that I think it is the method that he developed that we use until today, 90 years later. So this is how we raise flies in glass or plastic vials nowadays. So we offer them food that is banana, grape, or molasses based with yeast because they need yeast to develop properly. And you leave the flies there, they lay eggs, they will become larvae, impupate, and more flies will arise. What he did was adding watch glass here, something like that. So the Drosophila females, they lay eggs on this base here, that in our case, we do that in the lab. So it's a molasses-based gel, the thin layer of yeast paste. So they go there, eat this yeast, develop the eggs and lay, and we just go and count these eggs that were laid. It's the most accurate method until today to count eggs by Drosophila over time. And he was the first one to do that in this very accurate way. We are going to jump a few decades, go to the 60s. Actually, it seems like nobody wanted I don't care about it anymore. And there is this gap in the literature. Nobody studies temperature anymore. And then there comes this French guy called Jean Davy that is very famous. He passed away last year, actually. And he came with this ecological point of view and asking like, I want to know how temperature affects Drosophila in the nature. So he worked essentially with wild flies, like collected in the field, not laboratory ones. And he started, and he and his former students produced like dozens of papers showing many different aspects with many different species of Drosophila around the planet. And he contributed a lot. And he used some reproductive traits to analyze how the fertility was going, like number of eggs, hatching rates, and stuff, but nothing from the cell biology or molecular point of view. It was the 60s, so makes sense, right? We didn't have so many tools too. So we jumped to the 80s and 90s, and the people are still interested in temperature affecting fertility, but it's still from the point of view, from the ecological point of view, but also genetics, because now we have molecular biology growing exponentially in science. So people want to use this tool. So they want to know how the Drosophila are evolving, how the populations are behaving from the genetic point of view. But again, no cell biology involved or physiology too. Only more recently, from the 2000s, people started to be concerned, like how the gametes look like what is happening with the cells and the organs. So you can see papers. This one is from 2004, showing like, oh, if I raise the males in 31 degrees, how the sperm will look like. This is not the whole sperm, but this is the nuclei of sperm in a control fly. And if they are under heat stress, the nuclei morphology is out of place. So we started seeing this. Oh, stuff is happening. Or you could use the ovaries and see, oh, the ovaries are dying, and you're going to understand this very soon. But again, all these papers, they still are using too hot or too cold temperatures all the time for short periods of time. And it's interesting from the mechanical point of view. If you want to discover stuff, it's interesting. But if you want to have a more realistic point of view of what is happening, we should analyze more not normal temperatures, but more non-lethal temperatures, because those temperatures, they kill the flies. That's why they leave the flies for a short period of time, because they can't stay longer. And interestingly, I was already at Daniela's lab. This paper, with this brief communication, was published last year, bringing exactly what we were proposing at the lab. Look, survival is important, of course, but the temperatures, they can sterilize the flies with much lower temperatures. And that's the point that I just told you, like slides ago, like you can lay eggs, but if nothing's coming from there, doesn't matter. Even if the fly survives, what's the point? So they discussed that, like, you can sterilize males. In this case, they use Drosophila males only, but, and this can affect the whole system anyway. You don't need to kill them. So, and they propose, and I was very happy, there should be more studies using non-lethal suboptimal temperatures. That were exactly what we were doing. And I want to pause briefly. I have no idea about the time. 7, okay. So I need to do these parentheses because I've learned that you always should look at the history of what you're studying so you can understand what you're doing now, and also be thankful somehow. But even, thank these people that were before you. And this was the case of Robert King. Actually, Robert King, we know tons of stuff about Drosophila Genesis that I'm going to focus in the next slide. Because of this guy, his first paper was in 1956, and he analyzed many aspects of Drosophila Genesis using micrographies and drawings, with very detailed drawings. And in the 1970s, he published this compilation of papers with basic stuff that he described of about Drosophila Genesis. And if you add the keywords Drosophila reproduction at NECBI, and I don't believe it's your case, but maybe someone online doesn't know, but NECBI is this database of published papers that anyone can access, right? Doesn't mean that you're having access to the papers, but you can have access to what was produced anyway. And you see this exponential increase in the number of publications about Drosophila reproduction. And this starts around 1980s. And I believe that this is due to Alan Spradlin and Margaret Fuller work that were done in Drosophila Genesis and spermatogenesis, respectively. I am a scientific granddaughter of Alan's. Daniela was his mentee. So this is the work of them and his former students. So it's a field that is very active. very productive, both oogenesis and spermatogenesis, and it's still, we know a lot. There are a lot of questions that were not answered, especially from the point of view of physiology. So I think it would be nice to add these parentheses. So Drosophila Genesis. This is a picture that I took from a pair of ovaries. Each ovary has around 15 ovarioles. That is this guy here, this drawing here. And oogenesis is controlled by many physiological factors as hormones or gene expression. It can be controlled in many stages around the ovariole. We have regulation points around this piece of the ovary. And Drosophila ovary actually is a model for studying stem cell physiology. And Daniela was a pioneer in the study of the effect of diet and how stem cells behavior. So with this expertise, Daniela's expertise and the literature, our group now is interested in studying the detailed effects of chronic exposure of flies under sublethal temperatures, suboptimal non-lethal temperatures, so we can understand in detail how oogenesis is being affected. So explaining the ovariole, how this happened. You have this anterior tip that is called the germarium. It's followed by these developing egg chambers or follicles. At some point in the later stages of these follicles, the Telogenesis is going to start. That is the yolk uptake. So there is this oocyte here growing. We have cells that helps this oocyte to grow and develop until it becomes a mature oocyte, as in humans. So a sperm will fertilize this oocyte, a zygote will be formed, and you'll have an embryo, but this embryo is not in their bellies, it's in the egg. Once this embryo is fully developed, around 24 hours later, we'll hatch a larva that is like the child. In more detail, we have the germarium here that hosts the germline stem cells that are adult stem cells that gives rise to the oocyte as the ultimate goal, right? So in this part of the ovariole, they are dividing a lot, and we can analyze in very detail all these parts of the germarium. It looks like it's how we analyze this. It's how we study oogenesis. This is a fluorescence microscopy of the whole ovariole. This is the germarium is here, and using many genetic tools, we just can try to answer all the questions we have in mind. Okay, so this year, we published in the beginning of the year, we published this paper showing that warm and cold temperatures have distinct germline stem cell effects in Drosophila Genesis. And I'm going to show you like few data just for you to have an idea of how we can study physiology and cell biology and contribute to a more detailed knowledge to this field that there is almost none. Again, this is a plastic bottle. I showed you a glass, but we can use the plastic. We have vials, so we use vials or bottles to study the flies, subject them to whatever we want. In my case, it's temperature. So I had 3 incubators with different temperatures. They are all controlled temperatures, controlled humidity. So I just leave the flies there, take care of them. I dissect them whenever I want. And then the temperatures studied, again, it's Drosophila Melanogaster. So those temperatures are not random exactly. So 25 degrees is considered at the optimal temperature for Drosophila Melanogaster in the field and in laboratory. So this is our control. And we collect the adults. We are not working with larvae. We leave them at 29, that is warm. They survive, they lay eggs, or 18 degrees. that is chill, they are fine, they also lay eggs. And it's important to say that actually these two temperatures are very normal. It's normally used in fly labs because we use them exactly to take advantage of the genetic tools available. And the first question we asked is if they lay eggs differently under these temperatures. And yes, they do. Again, I need to remember you that I'm using adults. We already have data in larvae, but not in adults, and not using these temperatures. So this is the number of eggs that were laid per female for 20 days. So I counted daily the number of eggs using that method that I showed you some slides before. So the black line, it's always the control. The red line is always 29 and blue line is always 18. This is going to repeat in the other graphs. So it's going to be easier for you to remember. And doesn't matter if it's cold or warm, they lay less eggs, fewer eggs compared to the control. This is the very first question we wanted to answer. So this actually opened many other questions. Why? Why this is happening? What is impacting egg laying so much? And as I said, we can study many stages on oogenesis. I'm going to show you just a few of them. One of them is cell death. So we're here watching the germary again. As I said, the cells are dividing, the cells that are derived from the stem cells. We can use a dye to stain these cells that are dying, here in green. In blue is a stains for DNA, so we are seeing nuclei of all cells here, no specific cell, all cells. And in green are the cells that are dying. This could explain, well, if you have less oocytes being produced, let's think this way, you are going to have less eggs being laid. And what we found, actually, that cell death is temperature dependent. So in the cold, they die less compared to even to the control, that is the optimal temperature. But if you go to the 29 degrees, they definitely die much less compared to the control. So this could partially explain, at least at 29 degrees, why they are laying fewer eggs. It doesn't explain 18 degrees, but that's fine. It can partially explain 29. Another aspect that is very important to analyze is vitellogenesis. Why? Because it's where the yolk that the organism is producing is being captured by the oocyte. It's like the egg in the chicken. So the yellow part is the yolk. So it's the same idea. So eggs need this food source to develop inside the egg. So it's the same. So this is we call vitellogenic follicle because it's not yet fully ready. It's not a mature oocyte. The yolk is still being uptaken by the oocyte, but that's fine. This is only one stage we are analyzing. And if we see 18 and 25 degrees, they look fine. But if you go and analyze the nuclei, I forgot to tell you, but this is also staining DNA again. We see that we don't see as much vitellogenic follicles as we do with the other temperatures. With our trained eyes, we know that this follicle is dying. And if you quantify, as any good science must do, with good numbers, we'll have a graph, with robust results showing here, in this case, percentage of ovarioles with dying follicles. And we can see that if you compare blue and black bars, we don't see much difference in the numbers, but definitely we see a dramatic increase after 15 days in the death of these follicles. So something's happening, we don't know why, physiologically speaking, that is making these guys die under this temperature. Maybe, remember that I said to you, we can remove energy, reallocate energy to deal with some stress. So maybe the organism is saying, okay, let's get all this stored energy here because we need energy somewhere to deal with this thing we're dealing right now. So this is a very good idea. Actually, it's an idea that it's totally plausible to test. And the last aspect that we analyzed is exactly the hatching rates. So again, I'm here showing you this ovarial because we need a good quality oocyte, meaning a good quality sperm, so you can have a good, healthy embryo that will become a healthy larva. So our question is, does the temperature affect embryo development somehow? If it affects, how it's affecting oocyte and sperm quality. Now we are working on sperm, but we already have some data on the oocyte that was published. And I'm going to show you the main figure, one of the main figures of the paper, that is this one, the hatching rates. So here we have the percentage of hatched eggs analyzed in 4 different time points. Again, black bars shows the control. We see a decline in the number of eggs hatched that we attribute to aging. 20 days is a middle age fly, kind of. The blue bars was a surprise. Actually, the code improved the quality of the embryos that hatched. So definitely there was no decline, at least until 20 days, right? I don't know about after that. So somehow code is keeping these embryos good. And on the opposite, on the other hand, we have the red bars here showing like in 5 days, half of the embryos don't hatch anymore. So what is happening that 29 degrees, the flies, they survive, they lay eggs, but they're not hatching very well. Virtually, they don't lay eggs after 15 days. So those are all open questions to be tested from the cell biology and physiological point of view. They're open, and we hope to try to answer some of those in the next few years. So we propose this model here, showing that code actually has some positive effects on oogenesis, despite it decreases egg production. But worm normally has negative effects, besides, and also decreases the egg production. So I'm going to briefly talk about the mammals, just for you to have a more, you are more familiar with the mammals. So, and a bit about humans. As I said, no, I didn't say, but most of the data about the effect of temperatures on mammal reproduction comes from livestock. Humans, they are most epidemiological correlations, very few data about analyzing the biology itself. Most of the biology was done in livestock. We almost know nothing about the effects of hypothermia, but we know a lot about the effects of hot weather. So you can find tons of papers on livestock showing the effects of summer and winter in the livestock. There are a lot of them, and showing effects on lactation, fetal growth, quality of sperm, quality of oocytes. We have in vitro data, like you collect oocytes from the cows, subject them to 40, 41 degrees, and you analyze cell death the same way we analyze the Drosophila. And you see more cell death. Or you analyze in vivo, the cows in vivo. So you collect oocytes from the cows that were there in the summer, outdoors, and then using the same cows, you're going to collect the oocytes that were outdoors during the winter, for example. And you analyze if these oocytes will develop into embryos after in vitro fertilization. This is one example that I'm giving you. And you see that warm seasons, you have less embryos developing from after in vitro fertilization. So there are some molecular data using livestock as a model, very few on humans compared to livestock. And about climate change in mammals and human reproduction, that is the final slides. And I'm going to show you. Scientists are talking about climate change effects like for decades, actually, and they are saying, look, climate change is affecting the reproduction of organisms and the migration of organisms. So this is a 2003, 2004 paper, 2006 paper saying, look, we already have data, this is happening, and we don't know how, but this is happening. Nowadays, these papers are more recent. This is from 2019. This is from last year. They say, look, climate change impacts on fertility. That's a fact. We have tons of data showing that. And now the scientists are discussing from the evolutionary point of view, what is going to impact the evolution of organisms, because we are changing the environment. And this is a kind of pressure. So what is going to happen? And some scientists are discussing that now, using Drosophila also as a model. And in human reproduction, what we have, actually, we have very few data, but they exist and they are being published. Right now, this is from 2018. This is very interesting. They analyzed the census here in US from the 1950s, I think, until today. And they realized that women and men, actually, they didn't distinguish women and men, but they really realized that if they had unusually hot days in that summer or in that season, 19 months later, they saw a decline in birth rates. It's a correlation, of course, but they analyzed like tons of data, millions of people, showing that these exist. So there is some effect of these unusually hot days on the physiology of these people that is impacts the fertility of people months later. We also have this paper from last year showing that 1 degree Celsius increase in the residence of some women is enough to decrease the ovarian reserve on these women. But I'm showing only women because I just talked about oogenesis, but we have data on men too. It's not only women, we have data on men, lots of data actually. And last month, this review was released, actually showing the impact of climate change on reproductive health. One of the things is the heat, but they analyze other stuff like pollution, toxic chemicals and stuff. So again, this is real. Climate change has effects on reproductive health, not only fruit flies, but all organisms, it looks like. And actually, I want you to have these take-home messages. I hope it was a pleasant talk and you learned a lot of stuff. But if you live knowing that climate change worsened by human activities, increasing heat-related human deaths, suboptimal temperatures affect Drosophila fertility, and this is part of my work. Decreasing fertility due to temperature may be a major threat to biodiversity. Climate change is a major risk factor for mammal reproductive health. And this is the main take-home message, that basic science research can help bringing knowledge to public health actions. And actually, this is the main goal of the IPCC report. Like say, look, this is happening, we have suggestions, so we can try to deal with that. So, but we can't do anything, so it's up to you. And this only happens because we have scientists doing basic science in the universities and stuff. So, and I hope this was an opportunity to open a channel with you. This is my Twitter if you want to reach me out. If you have information that you think it's useful for me, I'll be very glad. And yes, I'll take questions, I don't know, online, if there is any, or if you have any, but thank you very much.
We'll ask the questions that are online, and I'll walk in and ask what you folks see.
Thank you. So congrats for your presentation, Anna.
Thank you.
If I can call you like that.
Yes.
So I am a clinical nutritionist and a scientist, and as it showed in one of your slides, you showed us that, you know, food intake, malnutrition can affect fertility. So could you assess food intake with these slides? And if the answer is yes, I'm very curious about how. Yes,
You can. Because these fly food, we are the ones, someone prepares, and it's somehow controlled food. So we know we prepare these recipes like a cake recipe. You have like, you put X gram, whatever ounces of this, whatever ounces of that, and so on. So we know the percentage. It has whatever percentages of molasses, whatever percentages of yeast. So you can manipulate the diet you offer to the flies. You can add drugs, you can add whatever you want. Is that,
Yeah.
you wanted to know?
This was not compounding variable in your experiment, like you could control that?
Yes, and thank you for asking. And also, I also measured if they eat the same.
Okay.
So, because if they don't eat the same, because how much you eat, also it will impact the number of eggs or the quality of this food, as I said. So they are eating the same, the temperature is affecting this behavior or not. So it's a good control for you to have in your work, if this is very important. Like,
Thanks.
yeah, we did that. But the food was the same in my case. Yeah, it was the same.
Other questions? Wondering, in addition to temperature, how hard is it possible to test temperature by CO2 concentration effects on reproduction?
Inside the incubator,
Yeah.
I don't control CO2 levels inside the incubator. I don't even know how to measure that. But no, I didn't. I only have the humidity controlled, but and the temperature, but not CO2 levels. But probably affects. I don't know how. I know flies are very sensitive, so.
Other questions? Oh,
So just FYI, I did South Culture a few years ago, you can't get incubators.
Yes. Yes.
My question though is, I know for genetic labs, so you must have looked at a multitude of genetic mutations in these flies that you narrow down to a pathway or a process using the genetics and molecular tools.
In my case, no, and we're doing, but there are papers showing flies that are more resistant to different temperatures, and yes, there are differences in the genetic. Actually, I can already partially answer you. If you change the genetic background of the same Drosophila Melanogaster, you already have different responses on Telogenesis, and I did that too. Actually, I used these white type, this yellow-white background, but I also tested 2 other genetic backgrounds, and you see differences. They behavior the same, but with variations. So definitely affects, but mechanisms, we're starting to look at this right now.
So along a similar line of questioning, if you look at flies that are exposed to higher temperatures in one generation and follow that through the generation, do you see similar modulation?
I never tested,
Okay.
but there are groups doing that.
other questions?
Yeah, I'm wondering if there might be a blessing in disguise here. If higher temperatures would have, and it would inhibit human reproduction, because I think that the a lower human population growth would be advantageous, and there seems to be no way of convincing people that population growth needs to be restricted. So wouldn't, if higher temperatures were to have that effect, wouldn't that be a benefit? Thanks.
It's kind of contradictory because I had a child and very recently, so no, I'm not advising you. And I don't think this is good because it's a very good question. Thank you for that, because this I can explain actually better. I don't want to say, let's increase the temperature so we have less humans, of course not. Fewer humans, of course not. But all this knowledge actually can help us to, as I mentioned in the very beginning, in improving methods and helping the fertility, not only about humans, but other organisms that may interest us, like disease vectors, crop pests and stuff. To control human population, I think there are other things involved that is not only temperature, involves, sharing resources more equally and stuff. So I don't think this is the case. I don't think increasing temperature is good. This is not the best way to avoid having children.
Ooh, crazy question.
Well, if this is the question you thought, yes, you can temporarily be sterile. Men can be temporarily sterile. If you go to saunas very often, and it's true, there are data showing like, actually, there are very recent papers that I didn't mention. I didn't have the time that I said there are papers showing effects on men. They has this very recent study that they analyzed men that work in very hot environments, like I forgot the name, but there are very hot, stuff, and definitely they have problems in the sperm. Of course, we can't just say, oh, it's the temperature, but it's a correlation again. But
my question is regarding insects, let's say in the tropics, like in Amazon, are they able to handle the heat better than like, let's say, insects in cooler climates like here?
Always someone asks this. The Drosophila, I don't know how the Drosophila there is adapted, but probably if there is Drosophila there, it's totally adapted compared to, I don't know, the Savanna. And two, they definitely are not, they can fly. And when the mean air temperature is 37 degrees, of course they can fly, but they definitely don't stay all the time in the air flying. They look for microenvironments where it's much, much cooler. So, and this question is so important that there are some researchers that instead of studying the chronic temperature as we do, they study changing the temperatures during 24 hours to see the impact on the fertility. So I could do that, but I'm just seeing the chronic effect. But you can totally, because
Another question.
here.
I'll be right back.
I was just wondering, is there any indication that this effect is reversible either in the flies or in mammals? Like, so if you go to a cooler temperature.
It is reversible in humans and flies. I don't know the max temperature in both, but definitely is reversible. You put it back to cooler temperatures, you give it time, and all the process restarts again normally. Yes, I did that too.
So that would argue that it's not a genetic change, or at least not exclusively genetic change. It's either transcriptomic or epigenetic.
Physiological, we don't know. My bets for now, it's physiology, only, metabolites, and I don't even know if it involves gene expression, but I'm betting my bet would be like the first approach, like the most fast response maybe is metabolic. I'm saying that because there are some metabolomic studies, but again, using these very extreme temperatures, so I don't know if it's real in my case. So, but doing a metabolomics it's an idea that we have in mind. It's there. Maybe we should do it.
So metabolomics, could you describe real quickly?
Oh yes, of course, this is not obvious. So you can grab the whole fly, actually, it depends. You can analyze whatever aspect of the fly organism. But in the case of fly, I would get his blood, the blood. If I would, I think I would start with the blood of the fly and treat this and put in equipment that will separate all the molecules that are inside this blood. So if there are dramatic changes in certain metabolites, and you collect the blood 25, 29, 18 degrees, and there are dramatic changes, you'll see these quantities changing. And so you can have a hint about the metabolites that are being regulated in these different conditions. I don't know if I was clear. Okay.
Yeah, it's parallel to genomics, proteomics, and metabolomics,
Yes.
and then viromics.
Yeah, thanks. I had one quick question first about one of your slides gave credit to the Drosophila workers of the world unite.
Oh, actually, this is a book that a researcher, Markenstein, I think it's Michelle, Daniela, I forgot. I think it's Michelle Markenstein. Sorry, Michelle, if you're watching this someday. She published this guide for Drosophila beginners with beautiful pictures and illustrations. So like, this is how a fly lab works. So, and the title is Drosophila Workers Unite. There's nothing related to unions.
Or politics, yes. Also, I wanted to ask, as a follow-up to my previous question, going back to the first two slides you showed.
The first, very first one
The
too?
very first, yeah.
Ooh,
Going back to all the
okay.
problems that
Okay.
have traced to increased temperature.
Oh, the CDC ones?
Right. I guess I go back to what I asked previously. I mean, all of these problems have their root primarily in an increased human population. So isn't there a need for some type of activity as far as population control? And I know this is a very controversial and unpopular question for me to bring up, but I'm convinced that unless the question of population growth can be resolved, all these other problems there will never be a solution for them. Thanks.
We could have a big discussion, a long discussion about it. I don't know if this is the time. But yet, I don't think increasing the temperature of the planet is a good method for birth control. I don't believe in that. And I don't think this is the main problem.
So, yeah. So over the years.
Yes, exactly.
Babies for women goes down.
If you educate women.
In developing countries.
Uh-huh.
Rate goes down.
Uh-huh.
Because they have fewer babies.
Mm.
More education they have, fewer babies they have. And that's like published in the finally in the IPCC. I haven't, but I know it's out there. And WHO has lots of statistics. So educating women around the world, in countries that normally don't allow it, one of the biggest things we can do.
You're relying all the responsibility on women.
No.
You should educate men too. Yeah.
Once there's no effective educating men. Any other question? Yeah. Thank you very much.